Fantastic. Okay. Well, hello everybody who has joined us this afternoon. Welcome to today's session, Car Tires and Salmon Health, State of the Science and First Nation Perspectives, featuring presentations from Dr. Jenifer McIntyre from Washington State University, Anu Rao and Graham Nicholas from Tsleil-Waututh Nation, and David Trout from the Nisqually Indian Tribe. We're very excited to be co-hosting this event with our Salmon-Safe US partners, and we're delighted to see another great turnout from a diversity of participants. So thank you all for your continued interest in this topic and for signing up today. My name is Teresa Fresco, and I'm the Program Manager for Salmon-Safe BC, and I'll be your moderator for today's session. I'm joining you today alongside my colleagues, Andrea McDonald, Dan Kent, Anna Huddle, and Brian Meggie from our homes on the traditional and ancestral territories of the Comox and Squamish nations, the Suquamish, Duwamish, Stillaguamish, Coast peoples, and the Cowlitz, Mutlama, Kathlamet, Clackamas, Tumwater, Tulalatan, Kalapuya, Wasco, Malala, and Watlala bands of the Chinook people in what are now known as British Columbia, Seattle, Washington, and Portland, Oregon. And I would like to invite all of us to acknowledge the work that Indigenous peoples along the Salish Sea have done and continue to do as the stewards and protectors of the lands, waters, and salmon for present and future generations. So next slide, Anna, please. I wanted to start with a quick overview of our proceedings for the next 90 minutes. I will be starting with an introduction to the Salmon-Safe program. I will then be passing it to my colleague Dan Kent to introduce our presenter, Dr. Jenifer McIntyre. Dr. McIntyre will present for about 30 minutes, and then we'll pass it to our First Nation and tribal panel for 30 minutes, and we will conclude with a Q&A segment. If you'd like to put forward a question to our presenters or our Salmon-Safe team, we encourage you to use the Q&A function, which you will find at the bottom of your screen. Next slide, Anna. This webinar is the third webinar of an annual three-part series called Salmon-Safe Communities Dialogues. We hosted our first webinar in May 2021, entitled Introduction to the Salmon-Safe Agriculture Program, and our second in March 2022, entitled Resilience, Salmon-Safe in a Changing Climate. Many of you will also recall our Car Tires and Salmon Health webinar part 1 that we hosted in February 2021. To access these and all our past webinar recordings, I encourage you to visit our Salmon-Safe BC YouTube playlist, which can be found on the Fraser Basin Council YouTube page. Just a note that we are recording this session today, and we'll be sending it to all of you in addition to uploading it to our YouTube playlist in case you'd like to access it again. We also encourage you to like us and our Salmon-Safe US partners on our respective Facebook and Twitter pages, and to visit our web pages, salmonsafe.ca and salmonsafe.org, to stay up to date on all Salmon-Safe news and events. Of course, this webinar series wouldn't be possible without the support of our BC funders and supporters who are listed here. We thank them for their generosity and for helping us make this work possible. So before we introduce our first presenter today, I did want to take a few minutes to provide an overview of the Salmon-Safe program. And firstly, to acknowledge that Salmon-Safe BC is hosted by the Fraser Basin Council. We're a nonprofit, non-government organization with a mission to advance sustainability throughout BC and a vision of social well-being supported by a vibrant economy and sustained by a healthy environment. So Salmon-Safe offers a peer-reviewed eco-certification program aimed at promoting transformative land and water management practices that protect and restore salmon health and watersheds. Salmon-Safe started in the United States in 1997 as a program of the Pacific Rivers Council in Oregon, and over the years, it grew into an independent, nonprofit, and eco-label. Salmon-Safe BC launched in 2010 with the agriculture program, followed by the urban program in 2013. And in addition to Fraser Basin Council, as you can see on the map on the right, Salmon-Safe is now delivered by a network of partner organizations along the Salish Sea. Salmon-Safe has a number of different certifications and accreditation standards as part of its broader program. These include our agriculture or farm certification, parks and natural areas, urban campuses, and infrastructure. Our firm accreditation programs include standards for construction, developer, and designer firms. The Urban Development Certification Program has 6 core standards of focus, including stormwater management, which will be touched on by our presenters today, water use management, erosion prevention and sediment control, pesticide reduction and water quality protection, biodiversity or enhancement of urban ecological function, and site climate resiliency planning. When a water course flows through a site, then 2 more standards are triggered, and these are in-stream habitat protection and restoration, and riparian wetland vegetation protection and restoration. The urban standards were developed in 2006 by a multidisciplinary team on both sides of the border, and were largely driven by the ecological need to protect salmon and our watershed ecosystems, as well as enhancing the visibility of those demonstrating leadership in these practices in the marketplace. And finally, I wanted to recognize our Salmon-Safe team along the Salish Sea, including Andrea and myself on the BC side, and our colleagues Dan Kent, Anna Huddle, and Brian Meggie from our US office. And with that, I'd like to pass it on to my colleague Dan Kent, who is the co-founder and executive director of Salmon-Safe, and he'll be introducing our first speaker today. So over to you, Dan.
Thanks, Teresa. Hello, everyone, and welcome to Car Tires and Salmon. I wanted to start with a shout out to Teresa and Andrea at Salmon-Safe BC, our co-hosts. And I also wanted to say thanks to the Boeing Foundation for supporting today's webinar in Salmon-Safe's broader efforts on the 6PPD issue. Salmon-Safe has been working for more than 20 years up and down the West Coast to build a market-based movement around water quality protection and habitat conservation in salmon watersheds. Across the Salish Sea, our implementation is guided by 2 high-impact organizations. South of the border, leading Salmon-Safe work with farmers throughout Puget Sound, Seattle-based nonprofit Stewardship Partners. And north of the border, as you heard from Teresa, she and her team at Fraser Basin Council leading Salmon-Safe BC. Salmon-Safe has tracked the great mystery around coho pre-spawn mortality in urban streams for many years. And when the landmark 6PPD research study was published more than a year ago, the first reporter that called the Salmon-Safe office asked, "So when are you going to be certifying Salmon-Safe tires?" Well, the answer to that question hasn't changed. First, we have to understand the science. And that brings us to Dr. Jen McIntyre in her presentation, looking at the state of the science around 6PPD. Today, it's an honor to again introduce Dr. McIntyre. She is Assistant Professor of Aquatic Toxicology at Washington State University. Dr. McIntyre's research specialty is the ecotoxicology of urban stormwater runoff. She focuses on the biological effectiveness of green infrastructure for preventing toxicity to salmon and other aquatic life. Her PhD work at University of Washington focused on the sublethal impacts of water pollution on fish health, including pioneering work on the impact of copper on juvenile salmon sensory systems. Welcome back to Car Tires and Salmon, Dr. McIntyre.
Thank you. Glad to be here, provide this overview. I'm going to pull up my slides here and get us going on the status update. And one more click for me. Here we go. Does that look right?
Looks great.
Okay, thank you. Okay, so I'm going to start off today with a recap of the issue, whereby we discovered the link between car tires and salmon deaths. And then I will move on to an update of what we've learned this past year-ish. So about 20 years ago, we published an article summarizing investigations into pre-spawn mortality of coho salmon that were returning to spawn in the Puget Sound area. This mortality was not explained by simple factors like elevated temperature, low dissolved oxygen, presence of pathogens, or the concentrations of commonly measured chemicals. Although we found evidence of exposure to common pollutants like metals and hydrocarbons, measured concentrations of those did not appear sufficient to explain the mortality. So instead, the weight of evidence pointed to the influx of stormwater runoff that was happening in concert with these mortality events. From stream surveys conducted by ourselves and many additional groups, including local municipalities, tribal nations, nonprofit groups, the researchers at NOAA Fisheries and U.S. Fish and Wildlife Service discovered that mortality rates in different basins throughout the Puget Sound were associated with roads and more specifically with traffic, and not so much with just imperviousness generally. So this let us know that within the realm of stormwater runoff, we needed to be looking at roadway runoff. And we subsequently discovered through empirical exposure studies that roadway runoff was sufficient to kill coho, and not just adult spawners, but also all of the free swimming life history stages. We have shown that this sensitivity of coho extends to juvenile coho, whereas juvenile and adult chum appear unaffected. In subsequent experiments with juveniles, we found that steelhead and chinook were the next most sensitive species of Pacific salmonids, and that sockeye appear unaffected, similar to chum. Now, with roads as the primary source of the mixture of chemicals that were killing coho, we focused on vehicle sources of pollution instead of trying to understand thousands of chemicals that were present there, many of which we didn't even know. In work led by researchers at the University of Washington, Tacoma, the PI Ed Kaloje, who I believe is here today, we may be able to rely on him to answer some of our questions. We looked at the chemical fingerprints of vehicle sources of pollution for 75 non-target organic chemicals. And those are shown here by these bands, either from the dark blue indicating not present to the dark red indicating highly abundant. And we compared those fingerprints, those chemical profiles with the chemical profiles of samples of roadway runoff, and also with samples of urban receiving waters, both of which have killed coho. So what stood out in the hierarchical cluster analysis is this one here. So this leachate made from tire particles. So the leachate is just water that's been in contact with tire particles and is able to pull out chemicals, so leach the chemicals from the particles into the water. The fingerprint for these this tire leachate was most chemically similar to the waters that killed coho. And this gave us a clue that tires might be the source of the acute toxicity. It's telling us that many of the chemicals that leach out of tire particles are present in these waters that kill coho. That did not necessarily mean that those chemicals would be responsible for the mortalities. So to learn whether the presence of these chemicals was in fact the source of the toxicity, we had to ask the fish. So we know that stormwater is acutely lethal to coho, causes certain pathophysiological changes, and does not appear to impact chum salmon, right, from our previous work. So through a series of experiments now, we determined that tire leachate it is sufficient to kill coho. So tire leachate within all of the chemicals that get into roadway runoff. Leachate, chemicals leaching from tires is sufficient to kill coho salmon. That it appears toxic in the same way as stormwater in terms of those behavioral and blood changes, and causing the same suite of behaviors, and that it affects coho, but not chum salmon. So narrowing it down. This then led to the discovery of 6-PPD quinone. And we started with this tire leachate, which contained more than 2,000 chemicals up here on the far left of the image. And we reduced the chemical complexity through a series of fractionation approaches. And this was led by Zhen Tian, who was a postdoc at the Center of Urban Waters, UW-Tacoma at the time. Now, through this process, we used coho mortality to track the toxicity through all of these fractions, some of which were non-toxic and The toxic fractions still contained our unknown chemical or chemicals that were killing coho. And we tracked this down to a fraction containing just 4 chemicals. And this was dominated by a single chemical that we now call 6-ppd quinone. We found that 6-ppd quinone, excuse me, in complex mixtures like roadway runoff and like tire leachate in the top picture here, in those mixtures, 6-ppd quinone was present at concentrations that do kill coho salmon. The red dashed line on these figures is showing the concentration of 6-ppd quinone that killed 50% of coho. This is called the median lethal concentration, or the LC50. And that value was similar both in the complex mixtures and when we just had 6-ppd quinone alone, which is the bottom panel there. So this indicated to us that the toxicity of the complex mixture containing 6-ppd quinone could be explained by the presence of 6-ppd quinone. And this made 6-ppd quinone our primary causal toxicant of this pre-spawning mortality syndrome in coho salmon. But 6-ppd quinone is not a tire ingredient. So how is it coming out of tires? It is derived from 6-ppd. And this is an anti-ozonant that is added to tires. It is an ingredient in tires. And here's the reason why. So UV reacting with nitrogen oxides, primarily coming from, in more built up areas from our vehicle exhaust from combustion engines, that creates ozone, ground level ozone. So it's naturally present gas, but it's present in higher concentrations where we have a lot of internal combustion going on. And this pollutant is highly reactive. So the ozone is continually interacting with the tire surface. It can penetrate up to a depth of about 20 micrometers into the surface of the tire. And there it can break tire polymers, all right, causing the tire rubber to crack. And I think we can all understand that would be bad for the integrity of the tire. So 6-ppd is there as a sacrificial molecule, all right? It's there for ozone to attack instead of the tire polymers. And there's enough 6-ppd in the tires that it slowly migrates to the surface of the tire to protect the tire rubber over the tire's life. Now, the byproduct of that reaction, or the transformation product from 6-ppd, is 6-ppd quinone. That's one of the things, as we'll go over here, that's made when ozone reacts with 6-ppd as it is designed to do. And this coincidentally turns tires this brown shade that you can see in this picture. You sometimes will notice it on the sidewall of tires, whereas you wouldn't so much see it on the tread because the tread is continuously being worn off to reveal fresh rubber. So importantly then, the 6-ppd quinone can leach into water directly from the tire surface or from particles that are worn from the tires. Okay, so that's the background. I'm now going to shift gears and talk about what we have been learning this past year. And this includes research now from groups outside of the Puget Sound region. I'm going to present the information as updates in terms of toxicology, chemistry, and solutions to this problem. All right, I'm going to start with toxicology, going over what we've learned about aquatic toxicity of 6-ppd quinone and mode of action. All right, so in 2021, a commercial standard became available for 6-ppd quinone. Standards can now be purchased from several companies, but we acquired it from HPC Standards. And when we tested it, we found that it was even more toxic than the 6-ppd quinone that we had made, either from 6-ppd using ozone or that we had extracted from tire leachate. There are two reasons for this. One was a higher purity of the commercial standard, and also the ability to correct for losses during the analytical process that weren't being accounted for. The result was that we revised the LC50 and our reported environmental concentrations down about 8-fold. So the LC50 was estimated at about 790 nanograms per liter, now is estimated at 95 nanograms per liter. In this past year, additional species have been tested for acute toxicity from 6-ppd quinone, whereas many species did not have an acute lethal response, all of the ones towards the bottom of this table. 2 more salmoniid species were found to be acutely sensitive: brook trout, which is in the Salvelinus genus, and another Oncorhynchus genus species, rainbow trout. Coho, not of course, but of course to me, coho are still the most sensitive of the organisms that have been tested. So 6-ppd quinone is very toxic to coho. How toxic is it relative to other toxic chemicals? I went through the chemicals for which EPA has established aquatic life criteria. These are concentrations of a chemical in water that the concentration of which is meant to protect aquatic species, all right? And to derive these aquatic life criteria, they're usually derived on the basis of the average of the LC50, that median lethal concentration, for the 4 most sensitive species that were tested. So if coho end up being the most sensitive species for 6-ppd quinone, I wanted to compare them to the most sensitive species that was tested for other chemicals. So I went to the data that was used to set these aquatic life criteria and extracted what was the most sensitive species that had been tested. And I'm showing here data for chemicals that had an LC50 less than 1 microgram per liter. And up in this list here is our 6-ppd quinone. So the other chemicals up at the top of this list are nearly all, and on this list in general, are nearly all organophosphate or organochlorine pesticides that have a neurotoxic mode of action. And 6-ppd quinone is among the most toxic chemicals that we now know of for aquatic life. We don't know yet if 6-ppd quinone is ultimately neurotoxic, but we do see effects in the brains of coho that are exposed to either runoff or 6-ppd quinone. Stephanie Blair last year published a paper in the Canadian Journal of Fisheries and Aquatic Science, where she explored the hemoconcentration, the thickening of the blood that takes place in coho that are made sick by exposure to runoff. This experiment focused on fish at this stage of where they lose equilibrium. She used Evans blue dye, which is a high molecular weight dye that will fluoresce under certain wavelengths of light. She injected this dye into the hearts of the fish and allowed it to circulate. She then rinsed out all of the blood. And you can see in this image that the gills are not dark red like they should be, or bright red. They are, skin colored, gill colored. Okay, and then she measured the fluorescence remaining in the fish's body. And the only fluorescence that should be there, so the only fluorescence that would be there is where plasma and large molecules, including this dye, had leaked from the vascular system into surrounding tissues. All right, now in this complicated figure here, there's just a lot going on, not that it's complicated. Panel C here is showing us the hemoconcentration of the fish that were exposed to runoff, right? This percentage of the blood that is cells versus plasma significantly increased for fish exposed to runoff. And these higher values suggest a loss of plasma. The images on the right here are showing the fluorescence of this dye in the head region of the fish, which Stephanie then quantified for the brain and the olfactory rosette, so two parts of the nervous system. And then those are quantified over here on the left. So relative to controls that were not exposed to runoff, there was clearly a lot of this dye that had escaped the vascular system and was left behind in these tissues. So vascular systems, or capillaries and stuff, are leaky to varying degrees, all right? And this is intentionally so in tissues like the liver. You want that to be leaky, allowing molecules to pass from capillaries into your tissues. But the capillaries in the brain should not leak, all right? Leaking in the brain suggests a breakdown of the blood-brain barrier, which is there to protect our brain tissues. All right, so she found the same thing for coho exposed to 6-ppd quinone. And ongoing studies are exploring both the severity and the timing of this blood-brain barrier disruption, and also extending this investigation into steelhead and chinook. Other ongoing toxicology studies in in our lab for 6-ppd quinone is to understand what environmental conditions affect toxicity for coho to start with. So for example, coho are exposed to waters both colder and warmer than those we typically are using in the lab. We expect that temperature will be positively correlated with toxicity, so that 6-ppd quinone will be less toxic at colder temperatures, if only because fish have slower metabolism at colder temperatures. We are additionally testing other relevant environmental and biological parameters, such as those I've listed here. Okay, moving on to chemistry. Here I will review global detections that have been presented in the literature and research into the transformation of anti-ozonides. So in this past year, studies have been published by groups in Canada, in Germany, in China, in Australia, where the samplings took place in those countries as well. And they were measuring 6-ppd quinone concentrations in runoff, in snow, in receiving waters, and also in soils and in dusts and in air, the very fine dust, I suppose, that's in air. And the results of these studies are in agreement with our expectation that we would find 6-ppd quinone everywhere that tires are in use. And the concentrations that they're reporting in these studies are also consistent with the environmental detections that we previously reported. So detections in runoff and snow adjacent to roadways are above the LC50 values. Detections in receiving waters tend to be near or above the LC50 in many cases. In the Puget Sound region, the UW team has been further exploring the presence of 6-ppd quinone in streams. In one study, they conducted a time trend in Miller Creek, which is a waterway close to the SeaTac airport, just south of Seattle. These creek waters were collected during 5 rain events with sampling every 30 minutes. So that's the time trend. Now the peak concentrations of 6-ppd quinone for each of those events, each of those 5 events were near or above concentrations that are expected to kill coho. Here I've overlain the median lethal concentration, that's that LC50, and also the LC25, which is the value that we would expect to kill about 25% of the coho that would be present. So, and we know from previous work that lethal concentrations don't have to be exceeded for very long in order to result in mortality. So for how long are they exceeded? In these 2 Miller Creek samples, here we have a storm in October and another in November. And reported here is the stream discharge in black with the axis on the right side, the amount of water moving per second, and the concentrations of both 6-ppd quinone and its parent compound, 6-ppd, on the left axis. So for these 2 events, the pollutograph, which is that shape and distribution of the concentrations, the pollutograph was more acute than the hydrograph, so suggesting that most of the chemical mass had washed off with the initial flush. The concentrations of 6-ppd quinone tended to exceed or equal 6-ppd, the parent compound, and also those concentrations were at lethal levels for several hours. And here I'm comparing that to that LC25 value. At other times and in other places, studies are showing that the pollutograph might extend well beyond the hydrograph, indicating continued mobilization of the compound after that initial flush of water of a storm. But also to note in this particular study, it's for a river in Toronto, so it's likely to have a lot of development around it. The concentrations of 6-ppd quinone were at levels lethal to coho throughout the entire storm. Okay, chemistry studies in the past year have also begun to, through chemistry studies in the past year, we've also begun to learn more about the transformation of 6-ppd. This study by Jimin Hu, who is a PhD student with Ed Kaloje, he explored the rate and the amount of transformation of 6-ppd that was exposed to ozone. So over time, you see it decreasing, and then you see 6-ppd quinone increasing. as ozonation proceeded. But only about 10% of the 6-ppd was transformed into 6-ppd quinone. So 90% of the mass that had disappeared of 6-ppd that had disappeared during transformation became something else. So they and other researchers have started to ask questions about these other transformation products. This study here is by a group in Europe, although Ed's group has a similar data set on transformation products of 6-ppd. So this diagram is proposing pathways of transformation of 6-ppd, and which I've circled in orange in the middle of this cluster here. And these are products that they observed in the lab through ozonation of 6-ppd, or also in field collected samples for various shadings and italic versus bold and things like that, from field collected samples of snow or water. So over here in green then is 6-ppd among those transformation products. And this extra panel is then the transformation products of 6-ppd quinone. What does it become as it further degrades in the environment? We are also learning about additional phenylene diamines that are present in the environment. These two studies sampled particulates from various regions in China. So in air samples on the left figure here, 6-ppd was present in all of these air samples, and that's the green bar. But the anti-ozonant 7,7-PD in the pink bar is very much dominating the phenylene diamines in these air samples. And then on the right, these are coarser particulates, so they're actually dust, and 6-ppd tended to be the most abundant of the phenylene diamines that were associated with dusts collected from roads and vehicles, so things related to vehicles and traffic. But in houses, that bar on the far right, the dust was dominated again by 7,7-PD. So what then are the risks associated with these phenylene diamines and their transformation products? That's some areas of ongoing work in the chemistry world. Okay, the final update here has to do with solutions to the pollution problem of 6-ppd quinone. I'm going to talk about treatment, and I'm going to mention source control. So green stormwater infrastructure is one solution to treating tire-derived chemicals and other stormwater pollutants. These are technologies that encourage runoff to spread out and to soak into soils instead of running directly into surface waters. So these are things that include permeable pavements and bioretention. And we study a variety of these at the Washington Stormwater Center, especially bioretention. And this is a technology that's used, for example, in rain gardens. This treatment approach uses an engineered soil to filter contaminants in the runoff as the runoff soaks through that soil. And in Washington, this is a mixture of, we just use a mixture of sand and compost. So we had previously shown that bioretention filtration can prevent the acute lethal effects of roadway runoff. These videos here are from an experiment where fish were exposed for 24 hours to either clean water, runoff, or runoff filtered through bioretention. The videos here are from just 4 hours into those exposures. So at 4 hours, the fish in the runoff in the middle video were already sick and they died soon after that. But the same water that passed through that bioretention filter system on the far right caused no acute effects for the fish. So we're showing that here for adults, but we have also shown this for juveniles and for alevin, freshly hatched alevin coho. We also have shown that it can prevent many of the sublethal effects we see in developing fish of various species. And finally, Ed Kaloje from UW has indicated that in the experimental bioretention systems that we have worked on together, that 6-ppd quinone is undetectable in the effluent coming out of the other side of that system. We have also worked with Federal Highways and the Washington State Department of Transportation on compost-amended bioswales, CABs for short, which can treat runoff adjacent to highways. Now, unlike bioretention, where runoff is treated by vertical filtration through the media, in CABs, the water is designed for primarily horizontal treatment, interacting with vegetation, and the surface layer of compost. So using retrospective analysis of water samples we collected for that study, we saw a high rate of removal of 6-ppd quinone from the in situ bioswales, though not as good as the removal rate that we saw for the bioretention systems. An ongoing experiment of ours includes looking at treatment of 6-ppd quinone in various depths of bioretention across time. These systems can be expensive to install. And how much media do we really need? We will soon complete the 10 simulated years of this study for treatment, and we look forward to sharing our results. A final experiment that I want to mention is led by PhD candidate Chelsea Mitchell in my department at WSU. She's been studying permeable pavements, among other things. So we know that permeable pavements are effective at removing suspended sediments through filtration and through sedimentation in the pavement pore spaces. Do they retain tire wear particles though? And do they do anything to mitigate the dissolved chemicals that are coming from those particles, right? Dissolving out of those particles. So in this study, we are looking for tire particles in the effluent, actual tire particles coming through that system. And we're also working with Ed Kolodje's lab to measure some of the tire-associated contaminants in those effluents. So ultimately, the goal is to replace 6-ppd in tires with an anti-ozonant that is safer for aquatic animals. And fortunately, this goal is shared by the US Tire Manufacturers Association and also regulators like the Washington State Department of Ecology and groups like Salmon-Safe. That's all I'm going to say on that topic at this time. So with that, I have completed the status update and we'll hand it over now to the panelists.
Thank you so much, Dr. McIntyre. It's really interesting to see all of the work that has been completed over the last year, and also some of the findings from that work. So thank you. And I know I see many questions coming into the chat. I do encourage folks who've just put their questions into the chat, if you could please use the Q&A box, which is located at the bottom of your screen, just allows our staff team who are tracking questions and comments to put that into their tracking form. Thanks all. And it's my great pleasure now to introduce our first speakers on our First Nation and Tribal panel, Anu Rao and Graham Nicholas, who are staff with Tsleil-Waututh Nation. So Anu Rao is a registered professional biologist, facilitator, and writer, and a senior environmental specialist on staff with Tsleil-Waututh Nation, focusing on marine ecosystems. Among other projects, she has been managing Tsleil-Waututh Nation's initiative in collaboration with the province of BC to update the water quality objectives for Burrard Inlet. For Graham Nicholas, with current graduate research investigates restoration opportunities for restoring culturally important wetland ecosystems at Tliches, Chatham Island, BC. Graham leads the Cumulative Effects Monitoring Initiative and Land, Water, and Environment field programs for Tsleil-Waututh Nation. So my great pleasure to pass it on to Anu and Graham.
Thank you, Teresa. I'm just going to share my screen here, if that's all visible. All right. Well, thank you very much for inviting me to present at this webinar. I'm going to give a bit of context as well as the bigger picture around 6-ppd quinone and how it fits into our work on water quality at Tsleil-Waututh. I work as a staff senior environmental specialist for the nation. So a bit of background: Tsleil-Waututh people occupied, governed, and act as stewards of their territory since time out of mind, and they continue to do so today. Tsleil-Waututh holds a sacred legal obligation and responsibility to protect, defend, and steward the lands and waters of their territory in accordance with Tsleil-Waututh law. This stewardship responsibility includes restoring conditions that provide the environmental, cultural, spiritual, and economic foundation for their communities to thrive. Burrard Inlet, which is this central inlet there, for those who aren't familiar with it, is central to Tsleil-Waututh people. Tsleil-Waututh's name for Burrard Inlet in their Hokkien language is Tsleil-Waututh. So Tsleil-Waututh literally means people of the inlet. According to Tsleil-Waututh oral history, knowledge, and archaeological records, approximately 90% of the Tsleil-Waututh diet was derived from the marine resources of Burrard Inlet and from Fraser River salmon. And today, the inlet's largely unable to support Tsleil-Waututh's needs. Adverse cumulative effects of colonial settlement, industrialization, and urbanization have eroded the ecological health, integrity, and diversity of Burrard Inlet. We've produced a summary of the contamination in the inlet based on research that we've compiled. And you can find that on Tsleil-Waututh's Sacred Trust website, which is linked there. Just go to the Research and Stewardship tab there. But in essence, we've synthesized that there's more than 600 point and non-point sources of contamination going into the inlet. 700 contaminants were detected between 1971 and 2016, and dozens of those exceeded benchmarks for water, sediment, and/or tissue. And those are just the ones we know of so far. In 2017, Tsleil-Waututh published the Burrard Inlet Action Plan, which is a plan to restore the health of the inlet. And the first strategy identified under its first goal is to update the water quality objectives for Burrard Inlet, which identify how clean the water needs to be in order to protect the most sensitive values. Tsleil-Waututh has led this work to update the water quality objectives in collaboration with the province of BC, leading to a first of its kind approach to water quality policy development. So Tsleil-Waututh priorities and values are at the foundation of this work and are driving it. And you see some of the products here, a collaborative policy document that's been developed. And these values include protection of human consumption of seafood, which is encompassed in the Tsleil-Waututh teaching, when the tide goes out, the table is set. And that sets a really high bar for water quality. The products of our work include so it includes these numeric and/or qualitative objectives for water, sediment, and tissue. We've also produced technical reports containing data analysis and recommendations, and we're currently preparing a report on contaminants of emerging concern. 6-ppd and 6-ppd quinone are included within this report. Additional products that we've created through this work, and the work overall reflects Tsleil-Waututh's holistic approach, which breaks down the boundaries and breaks down the silos that exist among other jurisdictions, and it looks at the inlet as a whole system. So as one example, we created this comprehensive map, as comprehensive as we could get it, of non-point sources of pollution. This includes tributaries, combined sewer overflow outfalls, stormwater outfalls, and the associated watersheds and sewer sheds around the entirety of Burrard Inlet, which was no easy feat to pull this thing together. And it's kind of a monster of a map, as you can see. In general, our pathway for proposing and adopting a water quality objective for a particular contaminant has been to seek existing benchmarks protective of the value that is most sensitive to that contaminant. These benchmarks had been identified by others following toxicological studies, again by others. What we've done is we've looked at the existing benchmarks, looked at the existing water quality data for Burrard Inlet, compared the two, and then proposed a water quality objective for a contaminant. But what do we do when either toxicological information and/or local data don't exist or are just beginning to be collected, like we're seeing through Jen's work, but that we know the substance is problematic? So what we've been doing is using a precautionary and conservative approach. So our current draft, water quality objective for 6-ppd quinone and other anthropogenic contaminants that have limited data and/or no benchmarks, is to see a decreasing trend over time in all media, water, sediment, and tissue. So this report, where we're mentioning 6-ppd quinone, it's still in its very first draft. It's going to be going through a review process involving Tsleil-Waututh as well as the province, and also a broad multi-sector roundtable. Some of the members of that roundtable I know are on this webinar today as participants. And that roundtable is a really key part of our work. This chart is showing its current makeup as of the other day. At our last virtual meeting, roundtable members expressed that they had never before experienced collaboration to the extent that they're experiencing as part of this water quality objectives project that Tsleil-Waututh is leading. And collaboration has occurred during all the phases of this work to date, from pulling people together and data together to developing the products and now moving into this implementation and action phase. And we can say that collaboratively contributed pollutants, such as 6-ppd quinone, require collaborative action for their elimination from the marine environment. And I'd say the forum and relationships that we've built through our work provide an opportunity to advance the conversations needed to reduce contamination in the inlet, including for emerging and acutely toxic contaminants. So here's my email if you'd like to follow up with me at some point. I apologize that I'm going to have to sign off pretty soon. I have another presentation at noon, but please do send me an email if you'd like more information about this work or to follow up. And I'm going to hand it over to my colleague Graham, who's going to carry on this dialogue about collaboration and Indigenous values in water quality research and management by talking about Tsleil-Waututh's field programs. So thanks again.
Wonderful, thank you, Anu. So I will just share my screen and get underway. So yeah, thank you for allowing me the time to speak today. My name is Graham Nicholas, and I am a senior environmental specialist, which I help manage the natural resource field programs. And so, yeah, as Anu said, Tsleil-Waututh's been on the land and water stewarding their territory since time out of mind, and the field crew is a big part of that stewardship today. It just takes on a different form than traditionally. However, we are still combining all that traditional ecological and local knowledge into our field programs, and a lot of the priorities are developed through the community and what the priorities are of that community. So I thought I would just show you a quick overview of some of our field programs. We are fortunate enough to have a dedicated natural resource field crew of six staff who we run, all year long, moving from one project to another. So this is just to illustrate where we are in the territory and sort of some of our core projects that we're doing. So we have a pretty big presence in Burrard Inlet. I'm only going to have time to speak about some of the water related projects that we have on the go, but our work takes us all throughout the territory of Burrard Inlet, Port Moody, up into Indian Arm and the Indian River watershed. So I kind of see this as we are in this huge data collection mode where we're really trying to understand the current state of Burrard Inlet as a whole, and all of the impacts, all these cumulative impacts of colonial development in the territory, all with the goal to understand and fill knowledge gaps to prioritize restoration and eventually improve the overall health of the inlet. As Anu said, Tsleil-Waututh people are people of the inlet, and 90% of their diet was from marine resources, and that has been more or less, there's a huge disconnect from that just due to all these cumulative impacts. So shellfish, great example, have not been able to harvest since the 70s just because it's too unhealthy. But we're working towards improving that, and things are improving. So yeah, quickly just to highlight some of our main core programs. So one of the big ones is with our CTD and Niskin program. So all those yellow dots are our sample sites, and we go to these sites every other week, and we have been doing that since 2019. So a really large data set and understanding of how this waterway changes from year to year. So we're really trying to understand the physical and biogeochemical aspects of this waterway. So with the Niskin sample, we take surface and samples from just above the seafloor and analyze them for some of these items that are listed here. Some more projects that we do on land are with the local creeks that run through the reserve, where we are monitoring and trying to understand, non-source pollution and point source pollution And there have been some pretty positive outcomes of this. And one of them is with that testing of, heavy metals and fecal coliforms. And through some of this, we were actually able to pinpoint that a housing development wasn't even connected into the municipal sewer line and was just, discharging into the groundwater and contaminating the water. So this is just an example of, through some of the crew's monitoring work, that we were actually able to, reverse some of this pollution coming in. And I mean, this is one of, hundreds of potential point sources, but, everything contributes and helps. So that is one of the positive things about this. So also 6-PPD became, with this research, we realized that that's obviously a huge concern given where we are located in this pretty heavily urban environment, a lot of similarities to Puget Sound and down there in Washington. There's a lot of roads, there's a lot of tires. So we were definitely interested in understanding, locally, the effects of the 6-PPD in Burrard Inlet. So I'm just going to quickly share 3 of the sites that have been part of this, and also to recognize that we've sort of just been scratching the surface of this, and our participation has only so far been in the data collection. We are more or less at capacity of fieldwork, so really like to stress the importance of collaboration. And so been collaborating with Dr. Tanya Brown at DFO, and she was, very timely able to share some preliminary results of what they've found. Now, this is not my slide, so I feel a little bit uncomfortable presenting on it, but I just wanted to highlight the fact that, we're trying to understand it, and it is present in some of these urban systems that feed into Burrard Inlet. McCartney Creek is the one that's closest to Tsleil-Waututh Reserve currently, and there are returning salmon, coho, and chum that go there. Very small amount that return, but this leads to the question of, what about a system that is maybe not so urban? And one of the main areas that we work in, and core importance and territory for the nation, is the Indian River watershed and the Indian River, more appropriately known as Inleilawatosh. And so this, in my mind, is one of the last remaining, quote unquote, wild rivers in the lower mainland, apart from the Fraser River, of course. But we are here in a system that hasn't been dammed. There's been, impacts in the past through logging and everything, but it's more or less remained wild. It moves and reacts like a natural river should. And we are getting fairly significant returns of salmon. So that's not a shadow, but a school of fish. And so last year we had about 1.5 million pink salmon return. Chum and coho are the other main species that return to the system. So, we get a lot of recreational users up in that watershed. Everyone has tires, so it's all contributing. And what does that mean for these remaining salmon populations that are already up against climate change and changing the worlds, that another stressor added to it? So this was from last year's pink run with the heat dome effects of some pretty big die-offs of salmon. So yeah, thank you very much for the time to share some of the work that Tsleil-Waututh is doing in their territory. So thank you very much.
Thank you so much, Graham and Anu, and thank you for all your work. It's just incredible to see the impressive scope of work that Tsleil-Waututh is leading, and I'm very heartened to see also the level of collaboration in that work. So thank you, and also thanks to Graham for staying on for our Q&A portion. So for folks that have questions, Graham will be here to answer them if they're directed to their staff. I have the great pleasure now to introduce David Trout, who will be our next speaker. As a Natural Resources Director for the Nisqually Indian Tribe, David heads a diverse department focused on salmon, shellfish, wildlife, and the environment in general. Throughout his career at the Nisqually Indian Tribe, he worked closely with the late Billy Frank Jr., the tireless advocate for tribal treaty rights and environmental stewardship. My great pleasure to introduce David. The floor is yours.
Thank you. And thank you, everyone. It's a pleasure to be here. I don't know about the 300 plus so folks that are in the audience, but I started out this panel sitting down, prepared to sit through my discussion, but about halfway through Dr. McIntyre's presentation, I had to get up. I had to get up and move around because this is urgent, and I am fired up after learning about the new research and what we're finding, and none of it is good. So I'm going to talk a little bit about the impact of all this on the Nisqually tribe and its treaty rights, and talk about some potential solutions and how to move forward with this. And so I will focus on how the 6-ppd quinone is negatively impacting our salmon populations and treaty rights. So the Nisqually Indian tribe is signatory to the 1854 Treaty of Medicine Creek, which promised in part the ability to fish as they had for generations in return for allowing the peaceful settlement of the Puget Sound region. Now the tribe and tribes have maintained their part of the bargain as peaceful settlement has obviously occurred, but unfortunately the promise to continue to fish forever has not been maintained. Salmon populations have crashed throughout Puget Sound Basin to the point where every major river system has at least one population listed under the Endangered Species Act, and in many places, multiple populations. Treaty fishing rights are illusory if there are no fish in the river. Salmon is the lifeblood of the Nisqually people. The current chairman, Willie Frank III, the son of Billy Frank Jr., compares his fishing time on the Nisqually to time spent in a church. And I've been out there with Willie fishing during Chinook season and coho season on the Nisqually. And while we're out there, he's constantly having a conversation with his dad, who passed away about 6 years ago, and his grandfather, who lived to be 104 on the Nisqually River. He's connecting to his previous generations of Nisqually tribal members while being on the water. And so being on the water, setting and checking his net, handling the salmon, and providing for your family is very much a religious experience for our tribal members. Today, many of our fishers are fishing the exact location where their great-great-grandfathers fished before them. With this in mind, we manage our fisheries carefully to ensure that enough fish get past our fishers so they can reproduce and provide for future generations. Around 20 years ago, when we were dealing with sharp declines in Chinook salmon and steelhead runs, the Nisqually Indian tribe developed a recovery plan, a science-based plan with the goal of restoring healthy, productive salmon runs that would support tribal and non-tribal fishing throughout the range of our fish. Our recovery plan included undertaking one of the largest estuary restoration projects on the West Coast, coordinating our habitat work with our hatchery programs, and changing the way we manage our commercial timberlands. These efforts have resulted in an increased survival rate for Chinook salmon and steelhead. However, we have not seen similar increases or response from coho salmon. Our fishing season, when I first started at Nisqually back in 1987, was 105 days long. In 2015, we only fished 8 days. We know that salmon survival, or lack thereof, is a complex mix of factors. But thanks to the gift of knowledge provided through the collaborative work between the University of Washington and Washington State University, we now know that 6-ppd quinone is highly responsible for the collapse of coho salmon throughout Puget Sound. This finding will help guide our efforts to recover salmon populations. 6-ppd quinone falls within the realm of the known and manageable issues affecting salmon. The immediate approach to solving this includes 2 key considerations. First, we need to remove 6-ppd from the source immediately by encouraging the rapid identification of alternative ozone preservatives that do not have similar or related impacts to the ecosystem. Candidates should be evaluated using the same techniques that were used to discover the problem with 6-ppd quinone. We also need to understand the impacts to juveniles and adults of all species of salmon and steelhead before it can be approved, used, and allowed to enter our ecosystem. Next, we need to identify and implement treatment options for stormwater, as mentioned by Dr. McIntyre, to capture and remove 6-ppd quinone from stormwater. We need to recognize that even if this harmful chemical were removed from tire production today, we will be dealing with the legacy of its use for at least the next 15 to 20 years. We need to be prepared for that reality today and commit to implementing and maintaining appropriate treatment systems. In the Nisqually, we're experimenting with scalable compost-based biofiltration systems on a state highway as a pilot project with one of our dear partners, Long Live the Kings, of which I see many of their staff are in the audience today. Shout out to Long Live the Kings. If we are successful, similar systems could be sized and retrofitted along all the roadways to treat for this lethal toxic chemical and also provide other water quality improvements. Key will be the long-term commitment to maintenance of these systems. The biofiltration systems, the rain gardens are only as good as their substrate is good, and that needs to be replaced and maintained, and we need to be on top of that. We as a country, as a region, have a history of taking bold actions to deal with these kind of toxic chemicals. In 1960s, it became apparent that DDT was the root of the problem for the very survival of our national symbol in the United States, the bald eagle. In the face of tremendous pressure from the chemical industry and large agricultural interest and operations, the EPA, under the leadership of my friend and mentor, Bill Ruckelshaus, banned DDT in 1972. The bald eagle, once on the verge of extinction, recovered over the next 25 years with successful delisting in 1997. A success story that should give us all hope when bold actions are taken. 6-ppd-quinone is the DDT of our generation, and we simply cannot wait forever to solve this deadly problem. The tire industry has suggested it could take or will take at least 10 years to find a replacement. That should be unacceptable to all of us, and we should all be fired up about this. 60 years ago today, our president, the United States, President Kennedy, challenged the nation to land on the moon by the end of the century. We landed on the moon in 7 years. It is inconceivable that it would take longer now to find a replacement that is safe for 6-ppd. Dealing with this significant and critical threat must be prioritized and integrated into all of our salmon recovery work. Removing this known killer will have positive impacts on coho survival, as well as far-reaching benefits to the region. I'm going to tell a quick story here that I had a phone call from a tribal elder at the Puyallup tribe, our sister tribe to the north of Nisqually. And he had known that I'd been involved in this discussion around 6-ppd-quinone and wanted to know more about it. So we discussed it. And by the end of the conversation, this tribal elder, who's also a fisher, was in tears. And he said, David, I don't know what to do. I'm a fisherman. I've been fishing all my life. And I now know when I get in my car and pull my boat with the trailer, all of which have tires, to fish my treaty right, I'm contributing to the problem. We need to deal with this now. And dealing with it will potentially avoid a listing of coho salmon under the Endangered Species Act in Puget Sound. It's been a candidate several times. It's still a candidate, and its populations are crashing. This might help avert that. More coho salmon will provide more food for our southern resident killer whales that are literally starving to death. More coho salmon will mean more fishing opportunities for recreational and treaty fisheries, supporting our small communities that are dependent on fishing throughout the Salish Sea. More coho salmon will mean more fish in the nets of my Nisqually tribal fishers, more fish in our freezers and our smokehouses, and more time on the river. And most importantly, more coho salmon will mean more time in the church of the Nisqually people. It's time to do something, and we simply can't wait. So I'm hoping all of you are energized and will go to action and make this happen. We need to find a replacement and treat stormwater better now for the benefit of fish everywhere. Thanks again for the opportunity to chat with you. I'll be happy to answer questions. Thanks.
Thank you so much, David, for your thoughts and pouring those thoughts from your heart and your energy and your call to action, that solutions are possible. And so I'm really looking forward to the Q&A and happy to have you. Thank you so much. So I'm now going to turn it over to my colleague, Brian Meggie, to facilitate the Q&A. And I know you all have been diligently putting your questions in the Q&A box, and I see also on the chat. So Brian, I will turn it over to you.
Well, yeah, if I had just turned my video on a split second sooner, I would have been standing up, because like how David had to stand up during Dr. McIntyre's talk, I just had to stand up there. And I really appreciate those words. And I know we all do on our Salmon Safe team, David, in terms of a call to action. And I'm definitely fired up, and I hope everyone else is too. So definitely am seeing that. I don't think we'll be able to get to all of these questions today. There are a ton that have come in, which is awesome to see. So what we'll do is I'll do my best to get through as many as we can. And then what we will do is we will compile the unanswered questions into an email with all the answers from our panelists, and then we'll send them out after the webinar. So with all that in mind, I will get started. Amy Patton asks, do you have any recommendations for a lab to test levels of 6-ppdq, and have you found differing results from differing lab methods? So that's a Dr. McIntyre question.
I mean, anybody who knows about this can weigh in. I'll share what I know, which is that I don't think there's anywhere in Washington State that is certified by the Department of Ecology yet to conduct those analyses. I know that Ed's group has an awesome method and is working hard to get that accredited. I do know that there's a lab in Sydney, BC, SGS Axis, that is doing those measurements on a commercial basis. And certainly other labs that have been doing this research are able to do measurements. Whether those meet, any regulatory criteria is not something I know.
Great. Next question going down the list: is 6-ppd quinone removed or detoxified by sewage treatment?
Okay, there is some research I didn't include today looking at 6-ppd coming into wastewater treatment plants. So it is observed going into wastewater treatment plants, which shouldn't be surprising, because it's a lot of, in some places, surface stormwater runoff will go into wastewater treatment plants. I don't remember if they did measurements after treatment in order to show whether it's eliminated. It will sorb to particles. It is likely that that a fair amount of it would get taken out with the sludge in the process of treating the sewage, but I don't know for sure.
We had several more questions kind of in the same family as this one, so I combined them all. Or we combine them all. Will increased numbers of electric vehicles translate into less ozone and therefore less 6-ppd quinone? I
really hope so. That's something I'm hoping will be, not necessarily me personally, but the scientists will be able to study and do some estimate quantification. Certainly ozone is present anywhere at ground level, but as I mentioned, UV emissions from combustion engines greatly increases those concentrations to levels that are dangerous for human health as well. So I think it will be very interesting to do an analysis ultimately when we learn the rates at which ozone is able to create the quinone from the 6-ppd in the tires, and get you an answer for that. I think it's a pretty exciting area. Obviously, electric vehicles have their own sort of environmental sustainability issues, but if we're just thinking about 6-ppd quinone here, it seems likely that would reduce the problem somewhat. Although then, on the other hand, electric vehicles, I believe, are heavier than internal combustion vehicles, and so that may cause more wear of the tires. So,
Definitely a lot to continue to research on that question, certainly. The next question, I guess, is for both probably, excuse me, Graham and Dr. McIntyre. Do we know why chum salmon are not affected by 6-ppd? And also, do we know why excuse me, I think I might have combined that. Did we just answer what the effects on humans are? Apologies.
No.
Yeah, so why are chum salmon not as affected by 6-ppd, and why are they not so affected by humans?
I don't know if Graham has any ideas. We don't know that yet. With our work trying to determine the mode of action, we're hoping that will lead us towards being able to identify what is it that's now different between these species for those target tissues. Let's say it's the vascular system, to be able then to answer the question of why there are these differences in species sensitivity. And in terms of human sensitivity, again, we don't know that yet. I don't know, but I sure hope there are people who are actively studying that on the human health side of things.
Great. This was another similar types of questions were coming in, so I combined it. What type of compost is being used in some of those permeability studies? And when is the permeable pavement study ending? And do permeable pavements actually remove 6-ppdq?
Okay, let's see if I can remember all three of those things. The type of compost, so far all of the compost studies that I've been involved with have used compost from Cedar Grove, which collects from at least the King County region. I don't know if they collect beyond that. So I believe it is a combination of residential, maybe commercial in terms of like restaurants. Not totally sure about that. I know very little about that, actually. So we do have some project work that will be starting up this year with support from the Washington State Legislature to look at compost use in bioretention, specifically focused on 6-ppd quinone treatment. Well, partially focused on that, but also asking the questions of, right, are there differences between different compost types? How much compost do we really need, given that it also contributes unwanted nutrients to effluent, at least for a certain period of time after it's installed. Okay, subsequent questions. Oh God, it was compost, and then it was gone.
When are the permeable pavement studies ending, and do they actually remove 6-ppdq?
Okay, let's see. So Chelsea is analyzing her data right now, quantifying those tire particles that are coming through the pavements and having the water samples analyzed. So I believe the study is done. The analysis will probably continue through this year. And so hopefully by next year, we'll have that out there for sharing. And so we don't yet know the answer to the question about whether it how it deals with particles and whether it treats the chemicals that are leached from the particles. We don't have those answers yet.
Right. That's why we do the experiments, right? Of course. Okay, cool. So moving on, does the soil being used to filter out 6-ppdq get adversely impacted? In other words, do the invertebrates suffer from adverse impacts from exposure?
We don't know yet. That will be very interesting to look at.
Definitely. This question is to any panelists. What is the best way to communicate the harm caused by tires to a science skeptical audience?
I think that you take the clip that Jen has of fish struggling to breathe and you show that to people, and it really drives home that this is a real thing and it's happening in our rivers every time it rains.
I think that's about as simple as it gets. We're visual beings, there's enough data to support that in science. So yeah, I think that would be great. Moving on, next question. How do tire sidewall protective products available to consumers affect the transformation of 6-ppd to 6-ppd quinone? And are those products known to contain 6-ppd?
I'm going to have to ask you to read that again because I was reading something in the chat.
No worries.
Question in the chat. I'm like, stop.
So how do tire sidewall protective products available to consumers affect the transformation of 6-ppd to 6-ppd quinone?
Ooh.
Are those products known to contain 6-ppd?
Oh, I don't know about those products. Not much of a car person. I did learn through learning about 6-ppd quinone that there are products that will clean the bloom off of your tires, the bloom being the 6-ppd quinone, which does provide a somewhat of a protective barrier then, just like when rust has already formed on a surface, less the rate of formation of more rust is reduced. But I don't know anything about other products. If anyone else has ideas, go for it.
It sounds like more to be done there, definitely. So moving on to another question for Graham, where exactly are the aquatic samples collected for measuring 6-ppd quinone? Did the recent highway flooding cause a major disruption to the salmon populations?
So all samples were collected up in creeks, freshwater, so far enough away from any saltwater influence. So that was just pure freshwater, and also collected after periods of drought and then right after that first flush. So to get any sort of thing coming off the roads. And then the flooding, we don't know yet, to be honest. The Indian River, certainly wasn't immune to the atmospheric river, and there's major washouts up in that system. So we don't know if last year's spawn has been washed out or not. We are currently trying to understand that through some of our other projects where we're tracking the outmigration of salmon. So we won't know yet, but we're working on it.
Great. I got another question in the chat for probably a couple other panelists, Dr. McIntyre as well. But what is the history of 6-ppd and 6-ppd quinone? When did it start being used in the tire industry, and what was it used for prior to tires? And then what sort of replacements has anyone been privy to?
Okay, and I'd be happy if anyone else here knows more about the history. What I've been able to learn so far is that it looks like there's no definitive, there's no like history of 6-ppd use in industry that's been readily available to any of us. But it seems like it started to be used maybe in the 60s, early 70s. And primarily it looks like it was replacing IPPD. Now, why it was replacing it, we aren't privy to either. Was IPPD more toxic itself? Because 6-ppd itself is toxic. So certainly it was known that those antioxidants could be toxic themselves, and IPPD was replaced. It sounds like almost entirely based on what the industry has shared with us by 6-ppd. Or was it a financial decision? A financial decision. It was more cost effective, or it lasted longer. Those are many of the factors that could be involved there. So I do want to go back. There's what is it? I wrote it down here. Encyclopedia of Industrial Chemicals, and that is renewed periodically over time. And a recent version of it that I saw included this quote that IPPD is increasingly being replaced by 6-ppd. And I want to take the time sometime to go back through the history of that encyclopedia and see when that statement first appeared.
Definitely. Yeah, that would be quite interesting to learn.
Yeah, I missed if there was more to that question.
Definitely.
I mean,
Yeah, and I guess the last part of that was also any
alternatives.
Yeah, alternatives that you know of.
Okay. Yeah. Well, there is the Washington State Department of Ecology and the California Department of Toxic Substances are both considering alternatives. What do we know so far about potential alternatives? I know the chat things keep popping up, but I keep reading them.
Block your eyes. Yeah.
there have been some assessment alternatives that have begun. Washington State Department of Ecology completed one recently summarizing what was known about 10 antioxidants that they had identified as possible considerations. And the U.S. Tire Manufacturers Association sent a letter based on that. Okay, Sarah is responding in the chat there. A letter explaining what they knew about some of those antioxidants and reasons for which some of those would definitely not work as a replacement for 6-ppd in terms of durability and that sort of thing, but identifying 3 of those compounds that there wasn't enough information for them to know whether it would or wouldn't work. But they could say that they didn't know that it wouldn't work. So it's a starting point.
Right.
And Brian, if I could add to that, and not specifically around a chemical substitute, but the idea of getting a chemical substitute, I think would be greatly aided if we were to establish a drop dead date of which this needs to be out of tires. If we said it had to be out of tires by 2025, I'd bet you everything in my pocket, which isn't a whole lot, but some, that we would find a solution. And so I think our elected officials need to lead on this one as well.
Definitely. Yeah, definitely. When put a little bit of pressure on, we tend to get some great things going, certainly. And this question, moving on to another question, just kind of showing how this issue is everywhere where there are tires. Is 6-ppdq being identified as a critical contaminant of concern in other regions of North America, more specifically such as the Great Lakes region?
I don't know what's going to happen there. Currently, I don't know what's happening in terms of regulations in other places in the country. I've never heard that. What we do hear a lot of is just information gathering, right? People making these detections like I was reporting today and working towards ultimately deciding whether they need to, set some regulations. Certainly in the Great Lakes, there are Pacific salmon, not native out there, but very, very popular for sport fishing, for example. And some of the species have established self-sustaining populations. Coho, though, not so much. They are a very popular sport fishery and they continue to be stocked in the Great Lakes. But it sounds like it has been difficult for them to establish any populations, which may have a lot to do with the amount of development that's in at least some of the regions of the Great Lakes.
And Dr. McIntyre, wasn't there a recent study that indicated brook trout are also susceptible? And so that should expand the map of concern as well, because they're everywhere.
Right. Well, and rainbow trout, rainbow trout or Oncorhynchus mykiss, which are our steelhead, and also inland rainbow trout. And rainbow trout have been introduced around the entire world and are very successful in very many places.
Yep.
Yeah, and that's great because that actually answers another panelist's prior question regarding if steelhead have been specifically affected. And of course, as they're genetically the same to rainbow trout, the answer is yes. So, and then and going back to this question relates back to something you said, Dr. McIntyre, about different types of tires, I believe, earlier in the Q&A. But in the absence of alternates to 6 ppd currently, do certain types of tires, harder, softer, winter, summer, more sidewall, less sidewall, et cetera, shed less 6 ppd or more?
Okay, so short answer, we don't know. We do know from previous research, just before 6 ppd quinone, people were concerned about tires and the chemicals coming out of them, and we're able to show, oh, this can be toxic to various aquatic invertebrates. Just whatever it is that's coming out of tires, and the mixture of chemicals. And those studies have certainly shown differences among individual tire brands and types and all those things in terms of toxicity. So I expect that there will be differences. And Ed and I would very much like to have funding to pursue, what is currently a best in class that people could elect to choose for a tire if they want to be part of the solution now.
Yeah, that sounds like that would be fantastic. So I'll just answer a couple more questions here just for the sake of time. And again, to reiterate, I will be compiling all of these later on and then sending them out with answers. So the next question is, has there been any collaborative discussions between government and/or indigenous nations to jointly draft legislation that could address this or water quality in general?
Well, the tribes down here in the Puget Sound region work through the Northwest Indian Fish Commission on issues that are held in common. And the commission is drafting letters now to key elected officials and to the EPA to try to raise the attention of those folks to this issue. At this point, we haven't gotten to the point of drafting legislation, but stay tuned, that might be in the works.
Great. The next question then is, how long do current permeable pavements remain viable, i.e. remain permeable? Like, is there, do they degrade over time? I think is what they're asking. Dr. Jen, you're muted. There you go.
Yes, handy Zoom tells me that too. So that's not an area that I know very much about at this point. Mostly I've been focusing on the bioretention side of treatment. And I know we have somebody in the chat who's also studies permeable pavement who probably can answer that question for us. So actually I'm going to not even start to speculate.
Great. Well, it's awesome to hear that we have folks actively working on it on this panel. So that's fantastic, or on this call. That's great. The last question that I will ask in the chat is, would decolonization efforts change the way we build streets in new land developments and replace existing neighborhood concrete high rises?
That is a great question. And I don't know the answer to that. I know that we're struggling with the impacts of growth in the Puget Sound region and the Salish Sea. And it's not just 6 ppd quinone, but it's a major problem, but all the other associated impacts. And I think we need to recognize that this is going to be something we're gonna have to wrestle with for a while as a region and answer the fundamental question is, can we live with a healthy environment, healthy communities, and healthy salmon populations? And I don't know the answer to that. I hope the answer is yes, but it's something we're gonna have to wrestle with.
Definitely. I guess I do have one last time for one more question. It seems. Do we know if 6 ppd is also used in bicycle tires and other types of tires?
Good thing nobody heard me swear just there. Yeah, great question and super interesting to know the answers, right? So I know that some of the ed students and postdocs have been actively working on that. I know they have a data set that they've put together looking at various, kind of household use items. And the other thing that I did specifically ask them about bicycle tires, and at least the bicycle tires that they had surveyed, this is Jimin in particular, it was not it was not detectable, I believe, is what he told me, which is very encouraging as a
Definitely.
bicyclist.
Right. Yeah, excellent. Well, that's all I think I have time for on the Q&A. Thank you so much, everyone, for sending in these great questions. And there's still quite a few. So as I mentioned a couple times previously, we'll compile those, get those answers, and then send them to folks on their email that they signed up for. So with that, I'll hand it back to Teresa, and we will go. Actually, I believe Anna, is the next one speaking. But anyways, thanks, guys.
Thank you so much, Brian. It's actually myself. So yes, thanks everybody for your questions. I also appreciate all of the messages in the chat and all of your respective expertise, you being able to share those. And so I saw that there was a request to save the chat. So maybe just a note for our team to save that, and we can distribute it to our attendees. And Anna, I'm just wondering if you could share our last two slides before we adjourn.
Yes, give me just one moment.
Thanks, everybody. Very lively chat there. Really appreciate all the sharing.
Sorry, Teresa, give me just one more second. Apologize.
I'm looking forward to going back and reading through the chat. I kept getting distracted by the things that were popping up. I had to move Brian's face to the top of the screen.
Yeah, no, it's great. And it's a fantastic community of people that have come together today. Anna, I'm seeing an email. Oh, there we go. Great. So once again, everyone, thank you so much for joining us. We had such a huge turnout. I think at the high point, we were at about 300 participants. Really appreciate you all using this afternoon and your lunch hour to be with us. We would love to hear your feedback about our webinar today, and we invite you to scan the QR code on the slide here or get in touch with us at salmonsafe@fraserbasin.bc.ca. Should you have any additional questions about the program, we encourage you to get in touch and check our website for further information. So salmonsafe.ca on the BC side, salmonsafe.org on the US side. And we've also put our contact information. So Anna, if you could flip to the next slide. If you'd all like to reach out with us, we've also added the BC and US email addresses. Happy to answer any questions. And we will also be in touch with you all following this webinar with a recording and also with some answers to those questions that were posed both in the chat and the Q&A box. So with that, I would like from my heart to thank our speakers and presenters today and our Salmon Safe US colleagues for all of the support in partnering to put this webinar on. And again, thank you all for joining this afternoon. So with that, I will call this to a close and adjourn. Have a good afternoon, everybody.